Very good morning to everybody. Welcome back to our daily lectures at New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. We are on the subject of molecular biotechnology at part 2 of the topic titled Molecular Diagnostics. We're at the subject of identification of specific antibody producing hybrid cell lines. The next task is to identify those hybrid cells that produce antibody against the immunizing antigen. One common screening procedure uses the culture medium, which contains secreted antibodies. The medium is collected from the wells that have growing cells and is added to a well of another microtiter plate that has been pre-coated with the target antigen. If the culture medium contains an antibody, primary antibody that recognizes an epitope of the antigen, it will bind to the antigen and not be washed away during a subsequent washing step. A second antibody, secondary antibody that is specific for mouse antibodies, is added to the wells of the test plate. It will bind to any primary antibody that is bound to the antigen. Before its use in the immunoassay, the secondary antibody is conjugated to an enzyme that can convert a colorless substrate to a colored compound. The presence of color in one of the test wells indicates that the original culture medium contained an antibody that was specific for the antigen, figure 9.4. If the culture medium does not contain an antibody that binds to the antigen, then the first wash will remove the primary antibody. Therefore, when the secondary antibody is added, it has nothing to bind to and is removed by the second washing step. In a well where such a sequence of events occurs, the substrate solution remains colorless. Those wells of the original microtiter plate, as media give a positive color response in the immunoassay, may contain a mixture of cell fusions. These cells are therefore diluted with culture medium and seeded into fresh wells to establish cell lines from single cells clones. After the clones have been cultured, their media are tested again to determine which cell lines hybrid spleen, myeloma cells or hybridomas produce monoclonal antibodies, molecules that recognize the target antigen. If more than one specific hybridoma is isolated, further tests are conducted to determine whether the different clones produce antibody against the same antigen determinant. Each monoclonal antibody producing clone can be determined more or less indefinitely in culture. In addition, samples can be frozen in liquid nitrogen to provide a source of cells for future use. Because a monoclonal antibody binds to a single discrete site, the specificity of an ELISA protocol can be considerably enhanced by using a monoclonal rather than a polyclonal antibody. Many monoclonal antibodies have been developed for use as immunodiagnostic agents. A variety of compounds and pathogenic organisms, table 9.2, as an alternative to isolation and synthesis of monoclonal antibodies in hybridoma cells and culture. Monoclonal antibodies and parts of antibodies, Fab or FE fragments, directed against the target antigen may be selected and produced in Escherichia coli. See Chapter 10. And table 9.2, which shows targets for diagnostic monoclonal antibodies, where you have polypeptide hormones such as chorionic gonadotropin, growth hormone, luteinizing hormone, follicle stimulating hormone, thyroid stimulating hormone, tumor markers, cytokines, drug monitoring, miscellaneous targets, and infectious disease. The biofluorescent and bioluminescent systems, proteins that naturally fluoresce or luminesce, or that can be easily induced to do so may be used as biological reporters in a variety of ways. For example, genes encoding these bioreporters proteins may be used to engineer cells to produce a measurable signal in response to a particular chemical or physical agent in the environment. In one version of this system, a gene for a fluorescent or luminescent protein is placed under the control of a promoter that responds to certain environmental signals, so that when the promoter is activated, a fluorescent or luminescent signal is produced. Color fluorescent proteins: green fluorescent protein, the 238 amino acid long protoprotein green fluorescent protein, isolated from the jellyfish Aquaria victoria, fluoresces green when it's exposed to ultraviolet light. While many fluorescent dyes are phototoxic, the incorporation of green fluorescent protein into cells allows intact living cells to be monitored in real time. The use of this reporter molecule has revolutionized fluorescent microscopy among its many uses. Researchers have used green fluorescent protein to monitor tumor cells in gene therapeutic protocols, to assess the responses of specific cell types, various therapeutic drugs and treatments, to monitor protein-protein interactions, and to monitor the fate of individual proteins in different therapies. Red fluorescent protein. Following the discovery and subsequent successful employment of green fluorescent protein, scientists began a search both in nature and by directed mutagenesis for other colored fluorescent proteins. Having multiple colored fluorescent proteins will enable several biological processes to be monitored at the same time. For the practical use of these proteins, it is essential that they both be as stable and as bright as possible. One problem with many of the naturally occurring colored fluorescent proteins is that they often have a tendency to form homodimers or homotetramers. Such multimeric structures can adversely influence the subcellular localization of the proteins, potentially leading to intracellular aggregation and other artifacts. One group of researchers isolated a gene for a red fluorescent protein from discosoma core and, by means of multiple random mutations, generated a mutant that existed exclusively as a monomer instead of a tetramer. With each iterative cycle of random mutagenesis, proteins that yielded red fluorescence that was both bright and stable were selected. The production of monomeric and red fluorescent protein required 33 mutations. This success notwithstanding, monomeric red fluorescent protein has several drawbacks compared to the native tetrameric form of the protein, including decreased brightness and reduced photostability. To address some of the above-mentioned problems with monomeric red fluorescent protein, as well as to expand the repertoire of available fluorescent proteins, additional rounds of mutagenesis were performed. First, DNA encoding 7 amino acids from the N-terminus of green fluorescent protein was added to the gene for red fluorescent protein. Then DNA for the 6 amino acids from the green fluorescent protein C-terminus was added to the gene for the red fluorescent protein. This construct then became the starting point for several additional rounds of random mutagenesis and directed evolution. Eventually, 7 different monomeric color fluorescent proteins were produced. It is argued that there is no one best colored fluorescent proteins. Some are brighter than the others, some are photostable, some are more sensitive to changes in pH. Thus, various fluorescent proteins may be used for different applications. Moreover, using 2 or 3 of these proteins, it is possible to label several different cellular components or cell types at once, thereby increasing the utility of this approach. Next topic would be luciferase. The luciferase enzyme, which catalyzes a light-emitting reaction, may be produced by a variety of different organisms, including bacteria, algae, fungi, jellyfish, insects, shrimp, and squid. Luciferase genes from bacteria are termed lux genes, while those from other organisms, the most widely studied and utilized being the firefly, are termed Luc genes. The Lux system includes 5 genes, Lux CDABE, and produces a peak of light at 490 nm. In some applications, all 5 Lux genes are utilized as a means of monitoring the presence and concentration of various compounds in the environment, such as organic compounds, including phenol, salicylate, benzene, trichloroethylene, ammonia, xylene, toluene, and ethylbenzene, or metals, including cobalt, copper, iron, lead, mercury, nickel, and zinc. When all 5 genes are used, the light-generating system does not require the addition of any other compounds. Therefore, following the addition of a contaminant-containing sample to bacterial cells carrying the LUXCDABE, a quantifiable amount of bioluminescence is produced within a period of a few minutes to no more than just a few hours. In some cases, the reporter system includes only the LUXAB genes. If LUXA and LUXB are together responsible for generating the light signal, the system requires that the substrate decanal be added during the assay procedure. The Firefly luciferase catalyzed reaction results in the production of a light at 550 to 575 nanometers. Moreover, the system requires the addition of the low molecular weight organic compound luciferin as substrate for the light reaction. Typically, LUX genes are used in conjunction with eukaryotic cells. Microbial biosensors. There is a need for methods that can easily and rapidly detect the large numbers of potentially toxic compounds that contaminate the environment. Once the contaminated sites have been identified and their range has been delineated, there are a number of highly sophisticated analytical techniques available to identify and quantify specific pollutants. Bacteria that are constitutively bioluminescent, that is, unlike the situation mentioned above, the bioluminescence does not need to be induced, are good candidates for pollutant detectors. In the presence of pollutants, the bioluminescence decreases, providing a clear indication of the presence of the pollutants. Naturally bioluminescent bacteria, such as the marine bacterium Vibrio fischeri, require saline conditions and a particular pH range and are therefore not useful for testing terrestrial groundwater. However, structural genes encoding enzymes that lead to bioluminescence, LUXCDABE, may be inserted into random sites in the chromosomal DNA of a soil bacterium such as Pseudomonas fluorescens. These genes do not contain a transcription promoter, so after insertion into the chromosomal DNA of Biofluorescence, the only luminescent colonies visualized in a dark room are those in which the LUX genes are inserted downstream from a constitutive Bifluorescence promoter without disrupting any important bacterial genes. The cells that luminesce to the greatest extent and have a growth rate similar to that of the wild-type strain and are selected for testing with various environmental pollutants. To screen water samples for the presence of various pollutants, both metals and organic compounds, a suspension of luminescent P fluorescence is mixed with the solution being tested, and after a 15-minute incubation together, the luminescence of the suspension is measured in a luminometer. When a test sample contains a low to moderate level of certain pollutants, the cell luminescence is inhibited, presumably because the pollutant directly interferes with bacterial metabolism. Since this procedure is rapid, simple, and inexpensive, it is a good first screen for assessing the presence of pollution at a particular site. After positive response with the bacterial biosensor, and actual pollutants can be determined by other methods. In the US, it's been estimated that there are approximately 87,000 different chemical compounds that need to be tested for estrogenic activity. That is steroid-like activity that can disrupt the endocrine system in vertebrates. While a number of different methods exist that could be used to test these compounds, they are too slow for this sort of large-scale screening. Therefore, scientists have developed a simple and sensitive system for the rapid initial screening of these 87,000 compounds. With this system, yeast Saccharomyces cerevisiae cells have been genetically engineered to produce measurable quantities of light in the presence of extremely low levels of estrogen compounds, as seen in figure 9.9. Using this method, in which luminescence is induced to test for estrogenic compounds, light production could be detected in as little as 1 hour. Moreover, following 6 hours of incubation, the assay attained maximum bioluminescence when the engineered yeast cells were exposed to as little as 5 by 10 raised to minus 11 M17 beta estradiol, a common estrogen. Of course, many estrogenic compounds require higher concentrations in order to be detected. The main drawback of this approach is that the yeast cell wall and transfer system facilitate the entry of some compounds into the cell and inhibit the uptake of other compounds. This can skew the results and in some instances may suggest that a compound is not estrogenic, when it is unable to efficiently enter yeast cells. Nevertheless, this technique is likely to identify a large number of estrogen compounds that were deemed non-estrogenic until they were tested with this protocol. At the same time, some groups of scientists are working to develop and perfect cells as biosensors. Others have focused their efforts on automating the system. Such an automated system might include genetically engineered cells that emit blue-green light at 490 nanometers in response to compounds, an environment that sustains the cells as light-tight enclosure and an integrated circuit with light detection and signal processing. While some simple prototypes have been constructed, the difficulties associated with utilizing living cells remain major impediments. And now to the topic of nucleic acid diagnostic systems. Nucleic acid, the genetic material of an organism, contains essential information that contributes to its various features and characteristics. For example, bacterial pathogenicity may be due to the presence of a simple specific gene or set of genes. Similarly, alteration of a gene may cause an inherited genetic disease in humans. In theory, the sequence of nucleotides that contributes to a particular biological characteristic is its distinctive signature that, if detectable, can be used as a definitive diagnostic determinant. Nucleic acid hybridization is the basis for rapid and reliable assays. The physical basis of these systems is precise nucleotide base pairing and hydrogen bonding between one string of nucleotides and a complementary nucleotide sequence. A general laboratory nucleic acid hybridization scheme is as follows. Bind single-strand DNA of the target to a membrane support. Add single-stranded labeled DNA to the probe under appropriate conditions of temperature and the ionic strength to promote base pairing between probe and target DNAs. And wash the support to remove excess unbound labeled probe DNA. Detect the hybrid sequences that form between the probe and target DNA. Excuse me. And now for these figures, please take a screenshot. We'll read the diagrammatic schematic representation of the strain of the yeast Saccharomyces cerevisiae that has been engineered to detect low levels of estrogen substances in the environment. Each yeast cell contains the human estrogen receptor HER gene integrated into its chromosomal DNA, one specific PUK, 404 that contains constitutively expressed bacterial laxity genes and flavin oxide reductase gene with the bacterium Vibrio harvey, and one plasmid that contains Lux A and B genes under the regulatory control of estrogen response elements and constitutive bacterial promoters is shown. Now, a nucleic acid hybridization diagnostic test that has been critical elements, probe DNA, target DNA, and signal detection. This type of detection system can be both extremely specific and highly sensitive. And the next would be hybridization probes. To be effective, a nucleic acid hybridization probe must have a high degree of specificity. In other words, a probe must hybridize exclusively to the selected target nucleic acid sequence. False positive, that is responses in the absence of the target sequence, and false negatives, that is no response when the target is present, severely undermine the utility of a diagnostic procedure. Probes can be specific at different organismic levels. This can distinguish between two or more species varieties, determine particular strains within a given species, or identify differences between genes. Depending on the requirements of the best protocol, probes can be DNA or RNA, long greater than 100 nucleotides or short less than 50 nucleotides, and chemically synthesized cloned intact genes are isolated to regions of a gene. Sequences that make effective probes can be isolated in a number of ways. For example, the DNA from a pathogenic organism can be cut with a restriction endonuclease and cloned into a plasmid vector. And recombinant plasmids are screened with genomic DNA from both pathogenic and non-pathogenic strains. Those plasmids that contain sequences that hybridize or lead to the pathogenic strain form the basis of sequences, for species-specific and even strain-specific probes. Additional hybridization tests with DNA from a wide range of organisms are then conducted to ensure that the candidate probe sequences do not cross-hybridize. Each potential probe is also tested under similar sample conditions, including the presence of mixed cultures, to determine its level of sensitivity. It is important to note that the knowledge of the genomic sequence of a large number of bacterial pathogens, currently several hundred, is facilitated the identification of unique stretches of DNA that could be used as probes. Just going back to diagrams that we need references and explanations. Right, so this is about assaying the presence of pollutants with genetically engineered bioluminescent Bifluorescence. You see that? Right, and another one diagram that talks about the various colored monomeric fluorescent proteins derived from monomeric red fluorescent proteins, showing wavelengths and color maxima. So there, one is honeydew, banana, orange, red, tangerine, strawberry, and cherry. Right. And here is figure 9.6, which is a construction of a modified monomeric red fluorescent protein, modified RFP. Take a screenshot and make notes, please. The regions of the gene encoding the NNC termini of the gene fluorescent protein was spliced onto the gene for MRFP following the removal of the portion of the MRPs encoding the N-terminus and the addition of oligonucleotide encoding a short linker peptide. This is a schematic representation of a bacterial cell engineered to respond to a particular environment compound, producing a detectable fluorescent or bioluminescent signal. This has been read to you already. This is a screening for the production of a monoclonal antibody. Spleen cells from a mouse that was immunized with a specific antigen are isolated and fused in culture with myeloma cells that do not produce antibodies. Few cells are selected for the ability to grow in het medium, which contains hyperxanthine aminoprotein, and aminotryptyrin, aminopterin, and thymidine. Cells that produce a specific antibody to the immunizing antigen. Hybridoma is identified by an immunoassay individually subcultured, or hybridoma which grows in culture and secretes a single type of antibody molecule is the source of a monoclonal antibody. So these are pictures. Please take screenshots. They should be of help to you. Okay, they're being shared with the figures through. Right. We finish up with that. Now we go to the next topic of diagnosis of malaria. An example of a diagnostic protocol that utilizes a DNA probe as a means of detection is a procedure developed for the detection of Plasmodium falciparum. This parasite causes malaria, a disease that affects about 1/3 of the world's population. The parasite infects and destroys red blood cells, leading to fever and, in severe cases, damage to the brain, kidneys, and other organs. Sensitive, simple, and inexpensive methods are required to identify the sources of the parasite in various localities, to assess the progress of eradication programs, and to facilitate early treatment. Currently, malaria infections are diagnosed by either microscopic examination of blood smears or immunological detection of parasitic organisms, antigens, effective but labor-intensive and time-consuming processes, especially given the large number of samples that need to be examined. All the immunological procedures for plasmodium detection, such as ELISAs, are rapid and amenable to automation. They do not always discriminate between current and past infections, because they are designed simply to detect anti-plasmodium antibodies in the blood of affected individuals. A DNA diagnostic procedure that selectively measures only current infections that are the presence of DNA-containing organisms was developed by using highly repeated DNA that is present in many copies from P. falciparum. First, a genomic library of the parasitic DNA was screened with labeled whole genomic parasite DNA. The most intensely labeled hybridizing colonies were selected because they were expected to contain repetitive DNA. The DNA from each of the selected colonies was then tested for its ability to hybridize with DNA from several other colonies, from several other plasmodium species that did not cause malaria. The DNA sequence that was chosen as a specific probe hybridized with P. falciparum but not with Plasmodium vivax, Plasmodium cynomolgus, or human DNA, despite the fact that P. vivax causes a less severe form of malaria. This probe can detect as little as 10 picograms of purified falciparum DNA or 1 nanogram of P. falciparum DNA in blood. More than 100 different DNA diagnostic probes have been isolated and characterized with the detection of various pathogenic strains of bacteria, viruses, and parasites. For example, probes that have been developed for the diagnosis of human bacterial infections caused by Legionella pneumophila, respiratory failure, salmonella enterica, blood food poisoning, Campylobacter intestinalis, gastritis, and enterotoxigenic E. coli gastroenteritis. Clearly, this is just the tip of the iceberg, because in principle, nearly all pathogenic organisms can be detected by this procedure. Detection of T. cruzi. The protozoan parasite Trypanosoma cruzi is the causative agent of Chagas disease. In this disease, the parasite invades the liver, spleen, lymph nodes, and several nervous systems, where they multiply and destroy the infected cells. T. cruzi is quite common in Latin America. It's spread by insects and is responsible for approximately 50,000 deaths per year. Diagnosis of acute Chagas disease is usually made by microscopic examination of a red blood sample. This lecture should be over in about a minute and a half from now, and the remaining portions will continue in the next lecture, right? Alternatively, a test that takes a longer time but ensures parasite has not been overlooked entails feeding a patient's blood to unaffected insects and then examining with a microscope the contents of the insect's intestines for parasites about 30 to 40 days later. Both of these tests are laborious, time-consuming, and costly. Chagas disease can also be diagnosed by immunological tests. However, these tests are notorious for producing false positive responses. As a possible alternative to these less than satisfactory procedures, several PCR-based assays have been developed at present. PCR assays for Chagas disease are used as adjuncts to the traditional diagnostic procedures that are currently in widespread use. In one of the PCR-based assay procedures, a 188 base pair DNA fragment that is present in multiple copies in the T. cruzi genome but absent in genomic DNA of several related parasites is the target sequence. So the presence of amplified 188 base pair DNA fragment is readily detected by polyacrylamide gel electrophoresis. In general, the minor variations in the methodologies, such as the primer sequences of PCR, can variations and methodology such as primer sequences, PCR, can facilitate detection of a wide range of bacteria, viruses, and parasites. Currently, there are various PCR-based and kits. We meet in the next lecture. Thank you.